Hi guys. So this one's going to be a pretty long one. We're just going to have to buckle down and do the best we can. This video is going to go over carbohydrates, lipids, and proteins, and I'm just trying to get it all in one video for you. You probably will have to pause so that you can get everything down after quite a few of these slides, so just kind of be ready for that. Okay, so we're going to start with carbohydrates. And carbohydrates is probably one of the macromolecules that you are the most familiar with because it's in the majority of the foods that you eat. So when you think carbohydrate, I need you to start thinking sugar, and I need you to start thinking of the fact that they're made up mostly of carbons, hydrogens, and oxygens. Okay, they tend to be composed in a 1 to 2 to 1 ratio. So for every 1 carbon, you're going to have 2 hydrogens and 1 oxygen. So if you think of the example of glucose, if you think of the example of glucose being C6H12O6, for every carbon we have 2 times the hydrogen, so that's where the 12 comes from. Then we have 1 times the oxygen, so that's where the 6 comes from. In terms of function, they're mainly concerned with energy storage, and they're also going to be your body's first go-to for fast energy. You're feeling low, that's the first thing your body's going to start breaking down to give itself an energy boost, which is why you crash so very hard after you've had tons of sugar or anything that has a lot of sweet in it. They also make up the raw materials for a lot of different things within a living system. Some organisms are going to use them for structural purposes, and there are a variety of things that we'll talk about in a little bit. They are made up of a small subunit known as a monomer, and those are going to be sugars. The kind of sugar depends on the type of carbohydrate that we're talking about. And some general examples of carbohydrates would be sugars, like the things that make your coffee sweet or your tea sweet, whatever. Starches, like what you find in rice and potatoes, and then cellulose, like what you find in plant cells. So let's talk about sugars with a little bit more detail. Now you always know when you're working with a sugar because it's going to end in OSE. The end of the word is always going to be an OS sound. We classify sugars based on how many carbons make up that particular sugar, so their prefixes will always be based in numbers. So for example, if you are a sugar that's made up of 3 carbons, then you're going to be a tri-OS sugar. Tri for the number 3, and OS for the fact that you are a sugar. If you are a 5-carbon sugar, then you're going to be pent-OS sugars. Pent for the number 5, OS for the ending, telling you that it's a sugar. And if you are a 6-carbon sugar, then you're going to be some kind of hexose, hex for the number 6, and OS for the ending, telling you that it's sugar. So that's one of the easiest ways to figure out what you're working with, because your sugars will always have that OSE ending. Now structurally, it just kind of depends. You can have anything from a straight carbon or straight chain type sugar that looks like this, or you can have ringed structures which look like this, and we'll talk about how you get rings in a little bit. So sugars have some simple units which we call functional groups, and depending on the location of these functional groups, it's going to identify what category of sugar you are working with. So we can break them down into 2 main groups, which are called aldehydes or ketones, and you know which one you have based on the location of the functional group, which is called a carbonyl functional group. I'm just write in a few notes for you that you'll be able to take down. So the carbonyl is your functional group. Okay, and your carbonyl, the location of your carbonyl is going to tell you whether you have an aldehyde or if you have a ketone. So aldehyde groups are always found at the end. Whereas carbonyl groups, or ketones, are found in the middle. Okay, so really the main difference between the 2. Now, in biology, form and function go hand in hand. You are going to do your particular job based on what you're structurally allowed to do. We can also change identity by just the simple movement of certain molecules. So if you look over here at glucose and galactose, they look pretty much exactly the same. They're both hexose sugars, which means that they are C6H12O6. But the difference between the two is what side the OH group is found on that fourth carbon. So in glucose, that OH group is found on the right, but in galactose, the OH group is found on the left, and that simple maneuvering of your hydroxyl group around that central carbon changes the identity of the compound. So in terms of structure, most of your smaller carbon structures, so your 4s and 3s, are going to find themselves in a linear formation where they're just pretty much in a line. All of the carbons line up one on top of each other. And they can be that exact same way if they're 5 carbons or 6 carbons as well. But in solution, your 5 carbon sugars and your 6 carbon sugars are going to form what we call ring structures. So the first carbon in your group, which would be this one right here, is going to bend around, and it's actually going to form a link between the carbon itself and the oxygen on this hydroxyl group on the 5th carbon. So this 6th carbon will still be in existence. It'll just sit on top of carbon 5, and we're going to get rid of some hydrogens and oxygens so that we can create a link right here. And that's how that ring structure is going to form. But it only does that in solution. Okay? So we go from this linear structure, that's what we call this one, to this ring structure here or here. Now it's going to be really important that you learn how to number these carbons. This is something that's going to be huge in terms of how we create these molecules and also how we name these molecules. So in a ring structure, we number the carbons starting to the bottom or to the right, depending on how you want to look at it, of the oxygen that's actually a part of the ring. So that would be this oxygen here. Everything starts this way. So that very first carbon underneath that oxygen is called carbon 1 prime, followed by carbon 2 prime, carbon 3 prime, carbon 4 prime, carbon 5 prime, and then if you have a 6th carbon, because you have a 6 carbon structure originally, that 6th carbon will sit on top of the 5th carbon. Now if it's just a 5 carbon structure, this will disappear. There'll be no 6th carbon. It'll just be this last 5th carbon in that ring. Now, knowing these numbers becomes important because when we start forming bonds, we literally use the name of the carbon that the bond is created in the name. So that gives you some information about what kind of bond it is and where that bond is located. So if you think back to the first slide in the video, or maybe the second slide in the video, I told you that carbohydrates, are all about energy storage, but it's short-term energy storage. So we're getting that energy by breaking down the bonds, holding the molecules to each other. So in cellular respiration, when we talk about releasing energy, we're releasing energy because we are breaking this bond that the energy is stored in. So we can also break our sugars up into groups based on whether they are simple sugars or whether they are complex sugars. Your simple sugars are going to be monosaccharides, so mono meaning one. They're made up of one type of monomer repeated over and over again, for example, glucose. You can also have disaccharides, which are made up of 2 different monomers, so for example, sucrose, and then you can have polysaccharides, which are really large polymers of repeating units, and a good example of that will be starch. So sugars are built through the process of dehydration synthesis reactions, and that's what's pictured here. If you think about the word dehydrate or dehydration, it means to remove water. So literally we get enough of the hydrogens and oxygens close together so that they bond and create water, and when they create water, the water runs off. Literally it'll leave the molecule, leaving these carbons available to bond. So you go from 2 individual molecules to 2 molecules that are linked together, right, like it shows right here. The kind of bond or the kind of linkage that holds them together is called a glycosidic linkage. So again, important that you know what that is. So this right here, glycosidic linkage. Right, so one more time, a glycosidic linkage, whoops, sorry, is going to be the bond that holds sugar molecules together. Right, so here's an example of another dehydration synthesis reaction where we're starting off with the monosaccharides glucose and fructose, and when we remove that water molecule, we are going to create a linkage between the two, and we're going to end up with the molecule sucrose, which is a disaccharide. And you know sucrose commonly as table sugar. Okay, so here we have, let's talk about polysaccharides for a little bit. And you know that polysaccharides means many, so literally this word means many sugars. And you get it when you link a lot of individual sugar monomers together. It's very simple to build. It costs almost little energy, not a whole lot to put those things together. It's still going to be held together by glycosidic bonds or glycosidic linkages. And it's just as easy to reverse the building process, so to break those bonds, and that causes a release in energy. So again, it goes back to the fact that carbohydrates are a very good store of short-term energy because it's easy to build them and hold that energy in bonds, but it's also just as easy to break those bonds and release that energy for whatever your body needs to use it for. Now, in terms of the function of polysaccharides, it kind of depends on what kind of organism we're talking about. If we're talking energy storage, then plants are going to store their energy in the form of starch. So again, think rice, think potatoes. In the form, if we're talking about animals, then animals, so us included, we store our carbohydrate polysaccharide form as glycogen. So starch in plants, glycogen in animals. And in animals specifically, that glycogen tends to be stored in liver and also in muscles. Now, if we're using it for structural purposes, then in plants, the most common polysaccharide is going to be cellulose, but in animals, it just depends on the type of organism. So arthropods, think crustaceans like lobster and shrimp and crab and stuff like that, or fungi, they're going to use a type of polysaccharide in a structural way, and that polysaccharide is called chitin. It's not chitin, it's just chitin. Chitin's pretty hard, and it makes a fairly good exterior or exoskeleton. If you think of stepping on like one of those things, you know how it'll make like a crunchy sound? It's making that crunchy sound literally because the chitin is breaking down. But that's how they will use carbohydrates in a structural form. So polysaccharides can take on two forms, and those forms would be linear and branched. So a linear polypeptide just means that you have all of them connected one to each other, and there's just one starting point and one ending point, or there are very few starting and ending points. So if you look on the diagram, so I can show you what I'm talking about. See right here how this is free? That would be one starting or ending point. And then up here, how this is free, that's another starting or ending point. Notice there's no other free molecule in this particular one. All the rest of them are connected. Okay? Over here, on the second molecule, we'd have a starting point here, and we have another one here, and another one here. But other than those three, the rest of the molecule is all connected one to the next. Now, when it comes to digesting molecules like this, you have to have a free end for your enzyme to work on. It can't just start in the middle. So on this molecule, there'd be only really one or two places to start. You'd have to start breaking it down this way, or you could start breaking it down this way, but you have to break it off molecule by molecule, which would take a long time. Same thing with this molecule over here. You could start on this end, or on this end, or on this end, but there are only three locations you can start from, and you'd still have to break it down molecule by molecule by molecule. So again, it would take a long time. So it means that your linear polysaccharides, because they take so long to break their bonds off, it's going to be a slower, more controlled release of energy. Now, if we look at animal polysaccharides, so in the form of glycogen, they're branched. Branched just meaning they have lots of different lines all connecting together. So again, let me see if I can illustrate what I'm talking about. So for example, here's a free end, here's another, here's another, another, here, one here, one here, one here, there's one here, and one here, and one here. So as you can see, way more free ends on this molecule than there is on these two molecules, because don't forget these are two separate molecules up here. So even though it still has to be broken apart like one individual sugar at a time, because you have so many more places to start from, the enzymes can break this molecule down a whole lot faster than they can here. Think of it as you'll only be able to have as many enzymes as you have free points. So on this molecule, we can only have 2 enzymes working at a time. Think of it as on this one, we can only have 3 molecules working on a time. On this one, it looks like we could have at least 10 molecules all working at the same time, which means this molecule gets broken down really quickly, and these not as quickly. So again, slow release of energy, slow and controlled, because those enzymes can only work 1 sugar at a time, and fast release of energy. So if you think back to when I talked about the difference between glucose and galactose, it's going to be something similar here. Polysaccharides have the ability to be diverse depending on what their molecular structure looks like. So your molecular structure is going to pretty much determine the identity of the molecule, and it's also going to determine its function or its ability to function in different circumstances. So hopefully you remember this term from chemistry, the word isomer. So isomer just means that we have the same molecular formula, C6H12O6, but we have different ways of making a structural form or a shape from that molecular formula. So if you look at these two molecules, they look exactly the same with the exception of where their hydroxyl group is found. Where the OH group is found, it's on the bottom of that first carbon on the molecule on the left, and then it's on the top of that first carbon on the molecule on the right. The difference between these two actually have a name. So this molecule is called an alpha glucose molecule, and this one is called a beta glucose molecule. They're both glucose molecules, but the difference in name comes from where that OH group is found. So if the OH group is found on the bottom, it's going to be an alpha formation. If it's found on the top, it's going to be a beta formation. So let's write that in. So this is our symbol for alpha, and that just means that the OH group is on the bottom. And it's going to be on the bottom of carbon 1. That's the one we care about. And then over here, the beta formation means that the OH group is on the top, and that is the top of carbon 1. Okay, so literally the only difference is the location of that OH group. Now, these two are called isomers of each other. They're actually isomers of glucose because they're both types of glucose, but there's a slight difference because of where that OH group is found. Why do we care so much? Because the alpha glucose formation is what we find in starch, and the beta glucose formation is what we find in cellulose. Again, why do we care so much? Well, most animals are very capable of digesting starch and almost incapable of digesting glucose, and it's because of the location of the OH group. Okay, so I've mentioned before that the reaction that forms these polymers are called dehydration synthesis reactions. The reactions that break them apart are called condensation reactions. Okay, so condensation reactions, this is the mechanism that's going to break up your carbohydrates, sorry. It's going to break the bonds between those groups of sugars. And all of this is enzyme controlled. So your dehydration synthesis reactions are enzyme controlled, and your condensation or digestion reactions are also enzyme controlled. So remember a couple of slides ago I said something about starches being easy to digest, and cellulose is a whole lot harder to digest, and for animals. And it's because of that kind of bond or linkage. So starches are made up of 1,4-alpha glycidic linkages, and we call them 1,4 because the bond occurs between carbon 4, let me show you, carbon 4 right here on one sugar molecule and carbon 1 on the other sugar molecule. So this is why I said knowing how they're named is going to be important later on. So every single one of these glycosidic linkages, that's where they occur. Carbon 4, carbon 1, carbon 4, carbon 1. So in our system, like in humans, for example, we have enzymes that can break this bond. And those enzymes work specifically with alpha glucose linkages. But we do not have enzymes that can break beta glucose linkages, which is why we cannot digest cellulose. We find it very hard to digest cellulose. If you think of eating celery, the majority of what you get out of celery is going to be water. The rest of it, we always term it as fiber or roughage, and it helps us with getting rid of waste, and it's literally because we can't break those molecules down. Okay, so when you think about it, cellulose is pretty much the most abundant organic compound we have in the world because it's in every single kind of plant. And it's also in a couple of different types of organisms that are not plants, so some of your bacteria and that kind of thing. Herbivores have developed a mechanism to digest cellulose, but the rest of us, not so much. Carnivores especially, we can't do it. We eat meat, and I say we because I consider myself a carnivore. Anyway, so we eat meat so that we can get our energy and our nutrients because our bodies are not capable of breaking down those beta linkages. So when you think of what certain animals eat, it's not just a matter of liking how that food tastes. That's kind of a human thing. It really boils down to I can digest this, I can get nutrients out of this, and it's readily available to me, so I'm going to eat it. As opposed to I cannot get all of my nutrients out of this one source of food, so I'm going to eat some other things that will help me get everything that I need. So cows, all they eat is grass for the most part, like if you left them alone naturally. And it's because they can digest cellulose, and because they can digest cellulose, they don't need to digest any other kind of sugars. They don't need to eat anything else because their bodies are getting all of their nutrition from the food they're actually eating. And if you think of gorillas, they can't digest cellulose very well, so they'll still eat leaves, they'll still eat plant material, but they're going to add to their diet by eating fruit or sometimes even meat. It just kind of depends. So herbivores can digest cellulose because they have a mutualistic relationship with bacteria. The bacteria that live in their gut is capable of digesting cellulose, and that's how those animals are able to get nutrients out of their food source. Now, yes, I understand that we also have bacteria that live in our gut, but our bacteria aren't as good at digesting cellulose, so it still doesn't really work out for us the way it does for herbivores. All right, so now we're moving on to lipids. So lipids are going to be very similar in terms of composition to your carbohydrates. They're still made of carbons, hydrogens, and oxygens, but they have long sections, what we call chains, of hydrogens and carbons bonded together. And we just call them hydrocarbon chains. So in this picture, that would be this region here, here, and here. If you look at it, it's just hydrogen and carbon, hydrogen and carbon. There are 3 different family groups of lipids, and they are fats, phospholipids, and steroids. And these guys do not form polymers, so they really don't utilize dehydration synthesis, except one group of them uses it to kind of put some pieces together, but they don't make traditional polymers the way that a carbohydrate would. So what do they make? They're just really big molecules that are made up of small sections, but they're not always going to be in a continuous chain necessarily. The only thing that tends to repeat would be these hydrocarbon sections. Okay, so starting with fats, structurally fats are made up of two main parts, a 3-carbon alcohol, which is called glycerol, and a fatty acid tail. Now, how many fatty acid tails just depends on what kind of fat we are talking about. So the fatty acid tail is a long tail of hydrogen carbons or hydrocarbons, and it gets the acid name because at the very front of the tail it has a carboxylic group, and carboxylic groups are acids in chemistry and in biology. So on this diagram, this structure right here, that C that's double bonded to an O and then bonded to an hydroxyl, that is called a carboxyl group, and it's an acid. Okay, so that's where the name fatty acid tail comes from. Now, to get these two pieces to join together, this molecule will undergo dehydration synthesis, and it'll do it for as many times as it needs a tail. So there'll be dehydration synthesis that happens right here to join this one tail, and then it'll happen another time and another time until the molecule has 3 fatty acid tails, and it looks like the letter E. So maybe in like biology freshman year, they always taught you that your lipids look like a letter E, and it's because this section of the E is going to be your tails, and this section of the E, so like think the backbone, that's going to be your glycerol. Okay, so when we make that connection between 3 individual fatty acid tails and an entire glycerol molecule, what we're building is a special type of glycerol called triacylglycerol or triglycerides. You'll have both terms interchangeably. So triglyceride is just made up of 3 fatty acid tails. So if you count them, tail 1, tail 2, and tail 3, and then they are linked to one big glycerol molecule. And this right here would be your 1-glycerol molecule. Now, the kind of bond that holds these together is called an ester linkage. And ester linkages happen between the carboxyl group on your fatty acid tail and the hydroxyl group on your glycerol. So this area that I have etched out like in a box, so here and here and here, that's where the joining is going to take place. That's where dehydration synthesis is going to take place. And that's where the ester bond or ester linkage is going to hold the fatty acid to the glycerol head. Okay, and then here's an example of that dehydration synthesis. So these purple blobs represent your enzymes. You're going to need an individual enzyme every single time you add on that tail. Okay, so let's talk function. So fats are very similar to carbohydrates in the fact that they store energy. And they store energy well because they're made up of these long hydrocarbon tails. Every single bond holding those hydrogens and stuff together is going to be a form of energy storage. Now, these molecules are nonpolar. They're nonpolar because they don't like water. They have no oxygen, which will kind of make them like water. And because they're nonpolar, it also means that they are hydrophobic. They're going to wrap themselves up so that they're not anywhere close to water because they really just don't like it. Again, in terms of function, they are energy stores, but they are concentrated energy stores. Every single bond between those hydrogens and those carbons to another carbon is an energy storage. They actually give you twice the amount of energy that carbohydrates will, but your body uses these for long-term energy storage. They also help to cushion your internal organs between your heart and your lungs and your liver and your spleen. You have small layers of fat that are there to prevent any major damage if your body gets jostled around. Now, if those fat layers get too large, then those create problems on their own, but a small amount of fat is a healthy thing because it helps to protect those internal organs. They also help to insulate your body. Now, not us so much, but if you think of organisms that live in really cold climates, so think of seals or whales that live in, the Arctic or the Antarctic, and they swim in really cold water, they have to have a way to keep their internal structures warm so that they can survive. And that's what blubber does. That's what fat does. It traps in that internal heat. So there are two main kinds of fats, and they are saturated fats and unsaturated fats. And I'm sure you've heard your doctor toss these terms around. Like sometimes they'll run a test for like your good fats versus your bad fats, and your doctor will tell you to adjust your diet to suit. So saturated fats means that every single carbon is bonded to a hydrogen molecule or bonded to something. So it's fully saturated with all of its bonds or all of its bonding places being used. It has no double bonds, no carbon to carbon double bonds. It's going to create a molecule that is long and straight and is easily packed one on top of the next. You find saturated fats mostly in animal fats, and they tend to be solid at room temperature. Now, these are your bad fats. These are the ones you really don't want to be consuming a lot. These are the ones that when your doctors run those blood tests, you want those levels to be lowest because they contribute to cardiovascular disease and to especially the form known as arteriosclerosis, where you get a contraction, of your arteries and veins because so much fat has packed itself into that tiny little space. Now, on the other side, we have unsaturated fats. Now, unsaturated fats means that all of their carbons are not bonded to hydrogens, that there are some carbon-to-carbon double bonds, and they tend to be found in plant and fish fats. They also tend to be found in vegetable oils, and these guys are liquid at room temperature. So because they're not straight, because of those double bonds, they have these bends or kinks, and it means that the molecules can't be packed in nice and tight with each other. So that prevents them from attaching and packing in to your veins and to your arteries. So this is why these are healthier for you than the saturated fats. Okay, another grouping of lipids would be phospholipids, and the structure of your phospholipids is they're also made up of glycerol, kind of like your triglycerides, but instead of having 3 fatty acids, they have 2 fatty acid tails, and they also have a phosphate group. Now you need to remember that your phosphate group is negatively charged, and that becomes important in a little bit. So I always think of these as having a head region and a tail region, and you always hear us talking about the head is charged in one way and the tail is charged in another way, and we'll talk about what that means in a little bit. So here would be the head region of the molecule, and then here is tail number 1, and here is tail number 2. And I think the universal way that we draw these in biology is they always look to me kind of like a balloon with two strings or whatever. So phospholipids actually have a split personality, at least that's how I like to describe it. One region of your phospholipid is going to be hydrophobic, while the other region is hydrophilic. So remember, those fatty acid tails are made up of hydrocarbons, just bunches of hydrogens and carbons bonded to each other. They do not like water, so they're always going to orient themselves so they're as furthest away from water as possible. Now, the head region, the phosphate head region, actually is hydrophilic. It loves water. No problem being around it. So they're going to orient so that the head is closest to water. So what you end up happening, or what you end up seeing, is a situation where your molecules can look like this, where all the tails are on the inside. So again, they're far away from water. Not a very good artist, as you already know, but kind of get an idea. So this looks like the beginning of something that could be important very soon. So because of their dual personality nature, when you put phospholipids in water, the head regions are always going to orient so that they're closer to water, whereas the tail regions are going to hide from water. And they create these bubbles. Now, it's thought that these bubbles are your early evolutionary stage of a cell. This is kind of how it started off. Now, along with the fact that they orient in a specific way, if you put enough of them together, they'll actually form a double layer, which is known as a phospholipid bilayer. And that's what you can see here. Now, as you already know, your phospholipid bilayer is what makes up your cell or plasma membrane. And it all kind of started off like this. So this is huge in biology because your phospholipids create these sections or boundaries in water, and they define one side from the next, or an outside from an inside. And eventually it's to the point where they're used to make cell membranes. And we're very grateful for that because we're all made of cells, and without these cell membranes, the cell wouldn't be possible. The last grouping of lipids are called steroids, and steroids are ring structures that are fused together, and there are normally 4 of them, and then they'll have random different types of functional groups attached to sections of the ring, which will create different identities or different functions. So your main example, or the primary example that we always use for this, is cholesterol. And right, so this is the cholesterol molecule down here, and you can see 1, 2, 3, 4 fused rings, and then we have a variety of different things attached to them. And all of these different things are your different functional groups. So cholesterol is a really important part of a cell. It's a huge component of your cell, and it changes the ability of your membrane to function in a certain way. So it's found predominantly in the cell membranes of animal cells, but it's also really important because it's a precursor for all of the other types of steroids that your body is going to create, including your sex hormones. Now, steroids and hormones kind of go hand in hand. That's the structural form of them. So without steroids, you really wouldn't have those hormones that move around your body and tell different parts and pieces what it is that they're supposed to be doing. If you have high levels of cholesterol in your blood, it can also contribute to cardiovascular disease. So again, it goes back to that blood test that we talked about earlier. But in a certain amount, if you have a certain amount, I should say, then your body's functioning the way it should, because you do need some cholesterol. You just don't need an excess of cholesterol. All right, so let's look at it again. This is the structure. If we were to like draw it out by hand, this is what it would look like if we use molecules. And then if we put a 3D form to it, then this is kind of what the cholesterol molecule looks like. As part of the cell membrane, what cholesterol tends to do is it inserts itself in between sections of your phospholipids, and it helps to keep the membrane fluid. It prevents it from breaking. So if you have too much cholesterol, it makes the membrane very, very rigid, and if you don't have enough cholesterol, the membrane becomes very, very loose and it won't stick together. Alright, so again, remember I said that cholesterol is kind of your precursor to a lot of your different kinds of steroids. So again, let's look at that form and function thing. So here we're looking at different kinds of steroid hormones. And for a lot of these we have, let's think of an adult form, not an adult, sorry, but a male form versus a female form. So here, if you look at it, you can see the same 4 ring structures. 1, 2, 3, 4. 1, 2, 3, 4. The difference is, what else have we attached? So structurally they look very similar. Right here, the only main difference is there's this group here, and then here there's this group there, but other than that it's pretty much the same thing. Well, just the moving around of a few molecules, or the exchanging of one molecule for the next, can make a huge difference in terms of whether you're looking at a male hormone or whether you are looking at a female hormone. If you move to the image down here, same thing. Here we're looking at testosterone, which we always know we kind of associate with males, and here is estradiol or estrogen, which again we associate with females, and then there's progesterone. But look at the difference. Between testosterone and estrogen, it's just a matter of a hydrogen. That's literally the only difference between the two. And it completely, well, not the only difference. There's also some like double bonding and stuff. But for the most part, that's the main structural difference. And it definitely is going to make a difference between whether it's a male hormone versus a female hormone. Okay, so now we are on to the most diverse group of biomolecules, and they're diverse both structurally and functionally, and those will be our proteins. Pretty much when in doubt, it's a protein that's doing the job. They just do so much in your body. So they are used for enzymes, and we've talked about tons of them, and there are tons more to talk about, and we'll still not even talk about all of them as the year progresses. They're used for structure, for things like collagen and keratin. Collagen is a type of protein that's going to help with the fluidity and the resistance of your skin, whereas keratin is making up your fingernails and your hair. They are used as carriers and transportation molecules, like hemoglobin, which transports oxygen in your blood vessels around and around your body. They're used for communication in the form of signals, like hormones and receptors, where they accept those signals and interpret what those signals want and pass that message on to different parts of the cell. They're used in your body's daily defense in the form of antibodies. They help your body to move in the form of actin and myosin fibers that make up your muscles. And they're used for storage, not so much in humans, but in animals. Protein storage is huge. If you think of, oh, if I eat beans, then I'm still getting as much protein as if I ate chicken. Well, that protein is literally stored in that bean. So structurally, these guys do have monomers, and their monomers are called amino acids, and there are 20 different kinds. The polymer that these amino acids make up are called a polypeptide. And proteins can be made of one or more polypeptides. It just kind of depends. They tend to be large and complex molecules, and they normally have some kind of 3D shape to them.